Infrared radiation is part of the electromagnetic spectrum. It was discovered by William Herschel, who found that when he separated light from the sun into its different colours, beyond the red colour, invisible to the eye, there was some other type of radiation that caused the temperature to rise. Infrared radiation can cause bonds of molecules to vibrate more by stretching and bending them. The frequency of the radiation that causes the bond to vibrate can be characteristic of the bond. Consider the carbonyl group C double bond O. When infrared radiation in the range of 1640 to 1750 centimeters to the -1 is directed at a molecule containing the C double bond O, this radiation is absorbed by the bond, causing it to stretch. How the instrument works. A thin layer of liquid sample is placed between two sodium chloride discs. Sodium chloride is transparent to infrared radiation. A scan is made by varying the frequency of the radiation from 450 to 4000 centimeters to the -1. The different types of bond within the molecule absorb radiation at different frequencies, since the bond strengths vary for the different types of bond. When molecules absorb infrared radiation, not as much radiation passes through the sample, and so the response from the detector is low. In regions where the molecule does not absorb strongly, the response from the detector is high, as the radiation passes through the sample largely unaffected. A spectrum is then produced, where the wave number in centimeters to the -1 is plotted along the x-axis, and the detector response, or percentage transmittance, is plotted along the y-axis. The peaks within the spectrum show where the bonds within the molecule have absorbed infrared radiation. In AS chemistry, you only need to be able to recognize a few key peaks. The CH bond causes a series of sharp peaks between 2850 and 3100 centimeters to the -1. Consequently, most organic molecules show peaks in this region. A very important peak that you must learn to recognize is the carbonyl C double bond O peak, which gives a strong absorption of infrared radiation between 1640 and 1750 centimeters to the -1. You can think of this peak as a finger. Another important peak is one that results from the OH bond in alcohols, which occurs as a broad peak between 3200 and 3550 centimeters to the -1. The OH in alcohols can be thought of as being like a thumb. The peak that results from the OH group in carboxylic acids is much broader than the OH in alcohols and occurs between 2500 and 3300 centimeters to the -1. You can think of this as a cucumber. Let's use our toolkit. The finger for the carbonyl group, that's the C double bond O group that occurs between 1640 and 1750. The thumb for the alcoholic OH between 3200 and 3550. And the cucumber for the OH from carboxylic acids that occurs between 2500 and 3300. To distinguish between aldehydes and ketones, alcohols and carboxylic acids. Let's try with this compound. Does it have the finger? Yes, it does, which means it has a carbonyl, that is, a C double bond O group. Now let's check for the alcoholic OH with the thumb. No broad thumb-shaped peak between 3200 and 3550. Now let's try the carboxylic acid OH group. No very broad cucumber-shaped peak between 2500 and 3300. So our compound could either be an aldehyde or a ketone, but with our present toolkit we cannot distinguish between the two. Now let's try this compound. It does contain the finger, so there is a C double bond O carbonyl group present. The peak over here is too broad for an alcoholic OH. The cucumber peak corresponding to the peak for carboxylic acids fits, and so the compound is therefore a carboxylic acid. With this one we can see it has the thumb peak in the correct position, confirming the presence of an alcohol OH. There is no finger peak, and so no carbonyl group is present. Therefore the compound is an alcohol.